Hello, my name is Meghan Rettler, and today I'll be teaching you on synaptic communication using hand-drawn and candy diagrams. In a neuron, the cell body contains the nucleus and dendrites, and in this case, the nucleus is made up of a piece of chocolate, while the cell body and dendrites are made up of bubblegum. Then the cell body continues down to the axon, or the Hershey chocolate squares, which has a sheath covering for protection and insulation known as myelin. Along your axon, you'll find nodes of Ranvier, or tiny marshmallows, and also Schwann cells, the green M&Ms. At the end, the axon splits into several axon terminals, or more bubblegum, which create the synapse with another neuron's dendrite. When a neuron connects with another and shares information, it is known as synaptic communication. Action potential, or a signal of an electrical current, starts at the base of the cell body. For a resting neuron, the intracellular fluid is higher in potassium ions and has a resting point of -70 millivolts. The nodes of Ranvier are what are able to pass down the action potential down the axon. So here's a closer look at our marshmallow nodes of Ranvier along our chocolate squared axon in our neuron. And when a stimulus is introduced, it initiates an action potential to start gradually depolarizing. This is when the stimulus generates the action potential enough to open the voltage-gated sodium ion channels. The opening of this gate is called threshold, and the potential goes from -55 to -60 millivolts. Sodium ions rush from the extracellular fluid into the intracellular fluid until it reaches a +30 millivolts, and then the sodium channels close. The cell repolarizes and opens the potassium channels to move potassium ions from inside of the cell to outside. The action potential travels down axons due to saltatory propagation, meaning the flow of similar charges. The nodes conduct the action potential, and the local current skips the inner nodes and just jumps to the next closest node. Let's go ahead and take a closer look of when the action potential reaches the end of our bubblegum axon terminals. So here's a more detailed diagram. And when the action potential reaches the axon terminal, the presynaptic neuron will use the action potential to form neurotransmitters into his sacs, then dump them into the synaptic cleft via exocytosis. Once neurotransmitters reach the postsynaptic neuron, they bond to the ligand-gated ion gate and open the gate. The neurotransmitter bonds to the receptor and opens the gate by changing the receptor site's shape. This will then either continue or stop the impulse depending on the type of neurotransmitter. For example, an excitatory neurotransmitter will cause depolarization and continue the action potential, while an inhibitory neurotransmitter would cause hyperpolarization and stop the generation of action potential. The time it takes for this whole process to occur is synaptic delay.